Okay, so we are on chapter 4, section 2, and we're going to really talk about how protein structure is organized. And we're going to talk about how we go from that primary structure of just our sequence of amino acids all the way through to the three-dimensional shape that we end up with that really determines the chemical properties of the protein. And so what determines that 3D shape is really our side chains. So it's really important that you know those side chains of those amino acids. So that's my reminder to you to remember to study your amino acids. So we know that protein structure and function are really determined by that primary amino acid sequence, because that is what is going to contribute to how the protein is folded. And so we have two different interactions that occur in order to stabilize protein structures. So we have weak and we have strong. So the weak binding that we have is like hydrogen bonding, van der Waals, ionic interactions, hydrophobic interactions, things like that. Then you have stronger forces like disulfide bond bridges between cysteines, electrostatic interactions between our amino acids and sometimes metal ions, metal cations in solution. And that all helps to maintain our structure and to fold the proteins. So we know that proteins are not these rigid structures, right? We do crystallize proteins in order to see what they look like, but it's like taking a snapshot of someone, right? We are all moving and doing things and that kind of thing. And when you take a picture, we're only in one conformation, right? So you got to kind of think about proteins that way as well. Because if they're really held together by weak forces, well, then they can be flexible. So proteins are going to change their shape by breaking and reforming really weak interactions. And so those flexibilities allow it to have unique physical and chemical properties that are really pretty cool. All right, so our levels of organization of our protein structure, we have 4 levels. We have primary, secondary, tertiary, and quaternary. And that's what the rest of this section is going to be about, all of those 4 levels of protein structure. So we know primary structure is our linear amino acid sequence. When you are listing which amino acids you have from N-terminus to C-terminus, that's your primary sequence. And that's really going to determine how our peptide backbone goes from this linear shape into a stable folded 3-dimensional structure. And my arms can't move in all those ways. All right, so secondary structure. Secondary structure is an arrangement of our local regions of the polypeptide backbone. And so usually it's pretty regular and repetitive. So we have secondary structures that are very, very common. Beta strands, alpha helices, and beta turns. And the reason that we only have these basic 3 different structures is because there are physical constraints on these amino acids, right? Remember we talked about the phi and psi bonds on either side of your alpha carbon? And so those bonds are flexible, but only flexible to a certain degree, right? Because you have to maintain hydrogen bonding between the backbone to make all kinds of 3-dimensional shapes. So there is a restraint to what those phi and psi bonds can do. And so we represent the 3 different secondary structures as beta strands. So what you see here is a ribbon diagram, right? And so the ribbon diagram is generated basically from X-ray crystallography and NMR data. And then we see alpha helices. Alpha helices are like coils, like remember like the telephone coil, the retro telephone coil. And there are 4 basic kinds of helixes, and we'll talk about each of the major 4. And then we have beta turns. So beta turns are basically when you go from one beta strand to another beta strand. And those are the really organized 3 secondary structures. There's a 4th secondary structure that we call loops. Let's see if I can change the size of this. They're called loops, and they're not organized. and so we don't really call them as a secondary structure, but a lot of times you'll hear people talk about a beta strand, an alpha helix, a beta turn, and loops. Loops are variable regions of protein. They don't stick to a certain conformation or shape. And so there are really only 3, but people kind of talk about 4. So I want you to see that. Okay. And the reason why we see alpha helices, beta sheets, and beta turns are because they are exceptionally stable conformations. And we're going to talk about why they're stable. So what I want to do first is I want to show you, if I can click to the next slide, I want to show you a little animation of this secondary structure, because it's really hard to talk about without kind of seeing in 3 dimensions. So I want to show you this. So hang on, I'm going to see if I can make this go.
The 3-dimensional structure of a protein is defined by 4 hierarchical levels. The primary structure refers to the amino acid sequence of the protein. A protein's secondary structure describes the arrangement of local regions of the polypeptide backbone. Tertiary structure refers to the spatial location of all the atoms in a polypeptide chain. Finally, quaternary structure describes how different polypeptides are arranged within a complex. Almost all proteins contain cations of 3 tertiary structure elements: alpha helices, beta strands or sheets, beta turns, and loops. Alpha helices are right-handed helices that place the amino acid residue side chains on the outside of the helix relative to the polypeptide backbone. The alpha helix structure is stabilized by intrastrand hydrogen bonding between the carbonyl oxygen in one amino acid and the hydrogen of an amide nitrogen on an amino acid 4 residues away. Many alpha helices are amphipathic, meaning that one side of the helix is hydrophobic and the other side is hydrophilic. This property is important since alpha helices often need to interact with both the hydrophobic core of a protein and the aqueous environment at the protein surface. Amphipathic alpha helices are also able to pack closely together, with their hydrophobic surfaces facing inward or outward depending on the chemical environment of the protein. Another common type of secondary structure is called the beta strand. A beta strand is made up of an extended polypeptide chain where adjacent amino acid side chains alternate, being either above or below the polypeptide backbone. Multiple beta strands can form beta sheets through interstrand hydrogen bonds. Beta sheets can form through interactions of parallel beta strands or antiparallel beta strands. Beta sheets are rarely flat and are often twisted to various extents within the context of a protein. The third category of secondary structure found in proteins include beta turns and loops. A beta turn is a short 4 amino acid segment that connects 2 beta strands in an antiparallel beta sheet. Loops are irregular segments that often connect elements of secondary structure and are usually found at the surface of proteins. Through understanding the 3-dimensional structure of proteins, we can understand their function in the cell.
So what we're going to see is that those residues are held together a lot of times by hydrogen bonding, and that's what kind of holds everything together. So let's talk about some of these secondary structures in a little more detail. Let's start out with an alpha helix. An alpha helix, you have to kind of think about this. So this is typically a right-handed helix, and it is basically a ribbon that is wrapped or rotates around an imaginary rod that goes right down the middle, but it goes in a right-handed direction. So just like in the video, it was saying that it's stabilized by hydrogen bonding within the backbone, right? So we see this is really, really common in proteins because a lot of times strands of amino acids, so your primary sequence, will actually spontaneously fold into an alpha helix. And then once that happens, it's stabilized by those hydrogen bonds. And so when you look at a picture like this, what you're seeing is that the strand itself, right, here's the strand, this is the backbone of your peptide. And then what comes off of that are side chain residues. right? And so the side chains basically stick out from the ribbon backbone. All right, so what I want you to kind of see here is that these residues can actually pack pretty tightly together. They're only about an angstrom and a half apart. And so the reason is because how the backbone is oriented that it can pack in so tightly. So if we look at a chain of amino acids, right? So it's the same thing on the left as it is on the right. And what we'll see is that our carbonyl, right here, here's our carbonyl, it's our carbonyl oxygen, so it's specifically the oxygen, of a particular residue interacts with the residue 4 down. And it is the hydrogen, right? So it's going to be a hydrogen bond. So it's a hydrogen on the nitrogen that interacts with the oxygen, which is the hydrogen bond acceptor. And so it's 4 residues away. And so it allows for that twist so that those interactions can occur. So the average helix is about 10 to 14 amino acids, an average helix. And so you get about 3.6 residues per turn, right? And what's pretty interesting about this is you got to think about which residues this can actually occur in. And it's pretty good for just about any amino acid except for proline. So think about the structure of proline, right? Proline ends up as a ring that hydrogen, that bonds, not hydrogen bonds, but that bonds back to the nitrogen in the backbone. So the nitrogen in the backbone in proline lacks that hydrogen, and so it can't participate in hydrogen bonding. So when you look at an alpha helix, you're going to see no proline, which is actually kind of cool, right? So you got to think about it. If you have a mutation that results in forming a proline where there was no proline, that can be a really, really serious issue. Okay, so why, when we do this, do we end up with some larger effects on a protein structure? So if you look at how the amino acids are organized inside of an alpha helix, what you're going to see is that they do have, an individual amino acid has a dipole, right? And so it has one side of it that's positive and one side of it that is negative. And they're all arranged in the same way along the entire length of that ribbon helix, right? And so since they're all pointing in the same direction, right? All the negatives are pointing in the same direction. What you end up with is an overall helix dipole. Ooh, oh no, I did something wrong. Sorry about that. So, so what you'll see a lot of times is that this charge separation, where you have a partial positive charge on one end and a partial negative charge on the other end, is that it's stabilized by compensating charged amino acids at either end of the helix, right? So sometimes at the amino terminus, right, you're going to see charged amino acids like aspartate and glutamate, right? Here we'll have aspartate and glutamate, which are negatively charged, right, to balance out that positive. And then in the C terminus, you can actually have positively charged amino acids like lysine and arginine. And that will end up neutralizing the dipole of the alpha helix. There's another way that you can neutralize the charge, and you can neutralize the charge through interactions with other things, right? So things that are not inherent in the protein itself. So for example, water, right, is polar, so it can interact, phosphate, cations, anything that's charged, right? So what ends up a lot of times happening is that the N terminus or the C terminus is going to end up being on a surface of the protein so that it can interact. If it's a charged, partially charged N terminus or a partially charged C terminus, what you're going to see is it's going to interact with the environment because it's charged. So that's kind of interesting. And a lot of times when we look at these alpha helices, what we see is amphipathic. And remember, that means that one model is hydrophobic and the other side is hydrophilic. And so this actually allows the alpha helix to interact with, because all the side chains are sticking out, and you have to remember that all the side chains are sticking out. And so it can actually interact with the interior of the protein, which is hydrophobic, and it can interact with the aqueous environment that the protein is in. And so it's really important that you can stabilize your structure that way. And it actually facilitates folding, which is super cool. Which, by the way, these slides, I added a few slides to this chapter, and I forgot to post the updated ones. So I went back and I posted them to Moodle. So I know you are missing these slides, but you can go back and you can get them. So if you want to pause it and come back, you can. One thing is that if you want to know how an alpha helix can be amphipathic, what you do is you look at that primary sequence and you're looking for a interchange of hydrophobic and hydrophilic residues every about 3 or 4 residues. And so it makes sense that way, right? Because the helix is going to rotate every about 3 to 4 residues. So if you were to look at the initial sequence, what you're going to see is that you'll have, maybe in number 1, you'll have something that is hydrophilic, right? So it's on the face, it wants to interact with water. Now you're going to have a turn of the helix and you get to your second residue. Oh, now we're hydrophobic, so we're faced on the inward part of the molecule. Then we go to 3, we're still valine, we're still on the inner part. Then we go to 4, and now we're on the hydrophilic outer face. And so you can kind of track this. This is called a wheel, and it's amino acid wheel. And so it predicts what the ribbon structure is going to basically be like. And so if we continue to do that, what we end up seeing is that everything that is hydrophobic is on one side of the helix, and everything that is hydrophilic is on the other side of the sequence. And so when you model this in on the actual protein itself, what you can see is these yellow residues that I kind of highlighted in purple so you could see. These are the ones that are going to face inside. So these are going to be inside. And it kind of depends, like I said before in the last section, what kind of protein it is, what environment it is in. And then these hydrophilic are going to be on the outside if you're in an aqueous environment, right? Okay, so now beta strands. This is our next major class, and these form sheets. So you have one beta strand, and one beta strand with another beta strand with another beta strand is going to form a beta sheet. And so in this case, it is also held together by hydrogen bonding, but the hydrogen bonding is not within a single strand, it's between 2 strands. So one beta sheet in itself is not stable. One beta strand is not stable. Beta sheets are stable. So when we look at this ribbon diagram, what you have to always remember is that your arrow is going to point towards the C terminus, and the side chains, like an alpha helix, our side chains are sticking out, but they're not sticking out all over. They're either going to stick out above or below the peptide backbone. So they're kind of like up or down, right? And these residues are relatively far apart. So before in our alpha helix, we were about 1.5 angstroms apart. Now we're about 3.4 angstroms apart. So amino acids are about 3.4 angstroms apart. So they're a little more spread out, and one by itself is not stable. What ends up being stable is when we have sheets. So almost always, you're going to see beta strands in sheets. And the reason is that you're allowing for hydrogen bonding between the strands. And there are 2 different ways that beta sheets can form. They can form being parallel, or they can form being anti-parallel. So when we look at this, if you were just kind of looking at it, what do you notice about the hydrogen bonding? Over the same amount of space, here I have 1, 2, 3 hydrogen bonds. Here I have 1, 2, 3, 4. So the hydrogen bonding arrangement in an anti-parallel is more stable. So this one, so anti-parallel is more stable, right? And so what ends up happening is that anti-parallel becomes more common. You see it in more proteins. But you do see parallel, just not as often, not nearly as often. And so the other thing about beta sheets that's important to remember is that they're not flat. We want to think a sheet, right, is like a sheet of paper, it's flat, but it's not. It's twisted. And the reason it's twisted is because of all these side chains. If it was all lined up straight, right, all these side chains would be above or below, and they would actually start to impinge on each other, right? There's a van der Waals interaction that we don't want to get those nuclei too close. And when those nuclei get too close, we call that steric hindrance. And so they would start to want to twist. So instead of having that, what we have is the backbone kind of goes up and down and up and down and up and down, right? We call this a pleated sheet. If you're into clothing, this is kind of what it was named after, right? If you're making like a skirt or something that has pleats in it, it's not straight. It's actually got pleats in it, so it kind of goes up and down, right? So that's what it was named after. All right, that was a little side note, right? Okay, so most proteins are globular in shape, and the reason they're globular in shape is because we have lots of turns or loops to connect our beta sheets and to connect our alpha helices. So if you're connecting two beta sheets together, what you have is a beta turn. And so we have two different kinds of beta turns. We have a type 1 and we have a type 2. In a type 1, we have our carbonyl oxygen that's actually oriented inwards. So it sticks in towards the inside of the two sheets. So if you had two sheets here and here, then your carbonyl oxygen would be oriented inwards, right? If you're type 1, if you're type 2, that carbonyl oxygen sticks outwards, right? And let's see. Oh, and what's really interesting about this is that, see this carbon 3 and carbon 2, so if we're counting right inside the loop, carbon 1, carbon 2, carbon 3, carbon 4, what's really interesting is this is a really pretty sharp turn that these amino acids have to take. And so usually glycine is in either C3 or C2 in order to allow for that necessary angle and not have a bulky side chain that prevents that curve from actually happening. And so you need something really small in order to do that. And then what's going to stabilize it is once it makes that turn, then you're going to have this carbon 4, we're going to have a hydrogen bond between the nitrogen there and the carbonyl of carbon 1. So we have lots of stabilizing interactions there that allow that turn, once it's happened, to keep it stable. And that same hydrogen bonding occurs in type 1 and it occurs in type 2. All right, so loops. Loops are irregular. This is another slide that I added in. Loops are irregular segments. They range in length, 6 to 22 amino acids. And when we say irregular, a lot of people want to think that these loops are really flexible, and they're not. Other parts of the molecule are flexible, but loops tend to be very rigid because you have a lot of weak interactions between a lot of really, really closely packed amino acids. So they're usually on the surface. And the reason they're variable is so that you can have specific protein-protein interactions. Because if you have to have a repetitive pattern within an alpha helix or within a beta sheet, it doesn't give you a lot of variability in your proteins. But you want only protein X to interact with protein Y to make some reaction happen. And so how do you get that specificity is through variations in certain parts of the molecule. And so this is the loops that allow for particular side chain interactions between side chains on the loop of one protein and side chains on the loop of another protein. All right, so how do we know that beta turns and alpha helices are really common? So if you amino acids, if you plot those phi and psi bonds, what you can see is that we're going to plot phi and psi on either axis, and then we're going to basically, and this is just shaded, but they've done this on many proteins, and put like a dot where, okay, it's possible to have a phi and psi bond, right? That is this and here. And so what ends up happening is this area of this graph tends to be beta turns. You also have triple helices, but basically this big area is mostly beta sheets, beta turns, right? And then you have another big area right here, and this tends to be alpha helices. And we call this a Ramachandran plot, and it's after the person who figured it out. And you can see that although there are each angle, you have a lot of possibilities. There are only a subset of those that are actually possible for both phi and psi at the same time. And so this really determines what angles are really actually possible. Maybe we'll go to the next slide. Okay, so here are two examples. The first one is a estrogen receptor ligand binding domain. It's not the whole protein, but it's the ligand binding portion. And so if you did a Ramachandran plot for this particular protein, what you're going to see is a lot of phi and psi bonds that correspond to our helical region. So if we go back, and I erase everything I kind of drew on here, right? What you're going to see is that we have lots of alpha helices, not so many beta sheets, right? Now, if you look at GFP, our green fluorescent protein, we see a lot more in the beta sheets. And that's exactly what we see in the molecule. And so this has been done over many different proteins, and it ends up being true for like everything. It's really cool. All right, so we did primary, we did secondary, now we're doing tertiary structure. So our tertiary structure includes all the secondary structures in their folding pattern. So the average protein has like 300 amino acids. That gives you like 10 alpha helices, 10 beta sheets, and the rest turns and loops. And so the tertiary structure is the arrangement of those hypothetical secondary elements, right? How many alpha helices do we have in a row? You know, how are the beta sheets lined up? Parallel, anti-parallel. How many loops do we have? All that stuff put together is our tertiary structure. So, and I kind of said that, right? Oh, and the positions of the side chains, right? What side chains are hanging out where, what are they going to interact with, that kind of stuff. And so you can actually go online and you can go to the protein data bank, and they call it PDB, Protein Data Bank. PDB, if I could write one thing, PDB, right? So this contains basically a map of every protein that has been identified, and you can kind of search and you can go, you know, I'm looking for a common theme in a protein structure, and so I want to know what other structures maybe have that beta barrel like our GFP did, right? And so you can search through those by organism. It's a really cool database that you can learn a lot of information out, depending on what you're really looking for. Okay, so we have four major classes of structures, right? So if you look at A, the first one is predominantly alpha helix, right? So you have some molecules that have mostly alpha helices. Now you're going to see loops in there, right? Because you have to connect our alpha helices, but the primary secondary structure is an alpha helix. Then like our GFP, right? We had predominantly beta sheets. So predominantly beta sheets. Then you have molecules that are an alpha beta combination, combined. So alpha beta combined, that's what we call them. And in an alpha beta combined, you have a mixture of the alpha helices and the beta sheets. Then you can have a fourth kind of molecule where one portion of it is beta sheets and one portion of it is an alpha helice. And so they don't mix. And so this is an alpha plus beta conformation. So you have like all of your helical region here, and then you have that adjacent to your beta sheets, but they're not mixed together. So that's 4 different classes. All right, so we're going to talk about some tertiary structures that contain more than one structural domain. And they kind of basically are categorized that way because they are independent folding modules, right? And so let's look at one example. One example, this is a sorry, my phone is beeping. All right, this is a 4 helix bundle. 4 helix bundle. Oh, look, it's at the top. And so what you have when you look at this, right? You're going to look at it's 3 different views of basically the same thing. So A is our ribbon structure. And so we show the 3-dimensional parts. So we have 4 ribbons. It's kind of hard to tell though that you have 4 ribbons because it's arranged in its normal 3-dimensional shape. And this is the heme domain of cytochrome B. And so you can actually see the heme fit into a pocket inside of those 4 helices. And if you do a space filling, so B is space filling. So if you look at all the atoms and where they're taking up space, you can't really see the helices, right? But you can see those helices are packed in very, very closely, and that heme feels the only little pocket there was. So if you want to know how that's really organized, what you really want to look at is a topology map. Topology map, where you see, okay, I'm going to start at the N-terminus, and I have 1 helix and a loop, 2nd helix, a loop, 3rd helix, a loop, and my 4th helix. Then you can look at how those 4 helices bundle together. So it's important when you know your 3-dimensional structure, you can see it completed and you can see it before it completely folded into that 3 dimensions. So you can see your tertiary structure and in addition to your secondary structure. Okay, let's see. The next example is called the Greek key fold. And so this is beta sheets, right? So if you look, I think the easiest thing to do right here is the topology map to start out with. What you're going to start with is the N-terminus. So here's our N-terminus. We go into 1 beta sheet, then we have a loop, 2nd beta sheet, another loop, a beta turn. It's not a loop, it's a beta turn. Oh, beta turn. Come on. All right. Then we have another one, and then we go into our 4th beta sheet. And they call this a Greek key fold because it's very similar to the Greek key design that's found on some really ancient pottery. But when you actually look at the true 3-dimensional shape of a Greek key fold, what you're going to see is those beta sheets are not flat, right? They're bent in order to accommodate all kinds of 3-dimensional structures. And so it's really important to remember that those beta sheets are not flat, that they are curved into whatever shape they basically need. Okay, the next one is called a Rossmann fold. A Rossmann fold is where we have alternating alpha helices and beta strands. And you'll see this a lot of times in proteins that bind to nucleotides. And so you have these regions of alternating alpha helices and beta sheets. And what you can see is that some places you're actually going to have a skip. And so this is one big region, right? And here's a second region, and then those 2 regions are linked together. And so they fold in together to make a really compact structural domain. And those side chain interactions help to hold everything together. All right, now a Tim barrel. This is for super big proteins. This is really, really large proteins. A lot of times these proteins act as structural scaffolds. And so you'll find them in like metabolic enzymes. So you can help lots of different proteins go through sequence, and so it's a large domain, and so you can do kind of like a manufacturing line to make it happen. And this was actually named after a glycolytic enzyme, triose phosphate isomerase. So that's the TIM, triose phosphate isomerase. And it's just one thing to remember, that it's for large proteins. All right, the firm domain. This is a tertiary structure that's found in 4 different cytoskeletal proteins. Not any of my favorite cytoskeletal proteins, but in some of them. And so what's kind of interesting about the firm domain, and the firm, it was named, they just took a letter from each of the different cytoskeletal proteins, which I think was kind of not nice, but that's okay. So you have the FA domain. The FA domain is mostly beta sheets. And so if you look here, right, we do see 1 alpha helice, but it's mainly beta sheets. Your third domain, your FC domain, is also mostly beta sheets. And then your FB domain is only alpha helices. And so these proteins with these domains tend to bind to membrane-associated proteins, sometimes glycolipids, and they serve to really anchor proteins to the cytoplasmic face of the membrane. But what's really interesting, this is something really interesting, right? All these cytoskeletal proteins have this firm domain, but the proteins have less than 30% identity to one another, yet still contain these firm domains. So you can have very similar tertiary structures, but not sequence homology. And this was one of those big things we talked about kind of, I think, in the first chapter, that you can have things that are structurally the same, structurally similar, and have very different functions. And you can have things that have lots of homology that have different functions. And so it's not quite a particular, if it has the homology of sequence, then it's going to conserve the function. Not always. So it's always important to remember there are exceptions to everything. Right? Okay, so let's talk about how these tertiary structures can be stabilized. We know that they're stabilized primarily by lots of weak interactions. We talked about that already. But there are 2 pretty strong interactions that they can be stabilized by, and one of them is disulfide bonds. So we see that a lot in our tertiary structure, and remember that occurs when we have nearby cysteine residues that are oxidized. And so it's really, really common in small secreted proteins that are in oxidizing environments, right? They need to maintain their functional activity even though they're in really, really dilute aqueous environments. And so you see them a lot of times in toxins and in growth factors. The other really stabilizing strong interaction can be with metal ions, but your book didn't have, so don't forget disulfide bonds and metal cation interactions. Right, my favorite protein, actin, does interact with metal ions, and it controls a lot of its structure, which is pretty cool. All right, so we're going to move on to quaternary structure. So most proteins are composed of multiple polypeptide chains that are organized together in order to make your big protein complex. So when this happens, you can have multiple copies of the same peptide, or you can have single copies of different polypeptide chains. And so those different polypeptide chains come from different genes. But the overall number and overall arrangement of the chains is your quaternary structure. So it's how many you have and how they're arranged, and that makes your quaternary structure. So just like I said before, you can have homodimers, right? Homodimers is where we have multiple copies of the same peptide, but dimer meaning two, so we have two of the same, right? But you can also have a heterodimer, which is two that are different, right? Just remember, like homogeneous and heterogeneous, right? Hetero means different, homo means the same. So it contains, if it's a dimer, it really should say contains two subunits from different gene products. So if you think about this, you could have more than just a dimer. You could have a trimer. Trimers are really common. That's 3. You could have a tetramer. That's 4. You could have a hexamer. That's 6. Those are kind of the most common ones. But a lot of times when you have these different subunits, you have multiple protein-protein interactions at that interface that helps stability, right? And so you have non-covalent bonds that occur, or you have disulfide bridges that occur that hold that quaternary structure together. So when we get quaternary structure, we actually increase the functionality of our proteins. So it can provide structural properties that are not present in an individual subunit. So maybe you have something really short, and when we put it all together, it gains the function. One example of that is keratin. Keratin has relatively long alpha helical subunits, right? And they wrap around each other. And because they wrap around each other, and how they do that, and we'll talk about that in a second, tells you the property of it. So I don't know if you know, keratin is in your hair, it's in your nails, it's in hooves, it's in horns, right? And so depending on how it's organized will determine its strength, right? It can provide a mechanism for regulation, right, through conformational changes. So a lot of times we see this in signaling proteins. In signaling proteins, we have like a ligand coming from outside and binding to a receptor. And so when it binds, that receptor changes conformation and induces a downstream effect. You can also have conformational changes due to phosphorylation of those side chain residues, right? We talked about which residues could be phosphorylated and dephosphorylated. And sometimes when that phosphorylation or dephosphorylation occurs, you have a conformational change in the protein. You can also bring functional components close together. So think about big protein complexes. They can get together, right? If you make this big protein complex, you can synthesize something like maybe a really long hydrocarbon, right? And so you do that kind of assembly line fashion. This part of it's going to hook on this part, and then we hook on another, and then we hook on another, right? And so it's that assembly line. Everybody has their own specific job within a big molecule. Let's see. So let's talk about some of these different kinds of proteins. When we talk about quaternary structure, fibrous proteins, this is where you have lots of multi-subunit complexes. So keratin, silk, collagen. So let's talk about each one. Collagen is a homodimer. So this is a homodimer of 2 helical peptides. So you can see these are 2 helical peptides, right? And they're wrapped around each other. And we call this wrapping around. If you have something that's helical and you have 2 of them and they wrap around each other, we call that a coiled coil. It's a coiled coil. And so inside of these peptides, you have about a 7 amino acid repeating unit. And usually about the 1st and 4th residues are hydrophilic. I'm sorry, they are hydrophobic, not hydrophilic, they're hydrophobic. So usually the 1st, let me write that down, 1st and 4th are hydrophobic. And so it allows those 1st and 4th residues in different helices to actually line up, and then they can pack really tightly together. And then you have some covalent cysteine disulfide bridges forming. And so depending on how many of those cysteines you have will tell you the strength of your keratin. So in hair, for example, the collagen in hair has a lower percent cysteine than, say, the collagen in horns. Horns has the highest percent. It's like 18% cysteine. And so that's pretty high. So it makes it super, super strong. So when we look, each of our individual helices are right-handed, and then the coil dimer love that. And this is really kind of interesting, right? Because this is how perms work, side note, right? You can chemically treat hair, right, which has lots of keratin in it, with reducing agents. So a perm will break the disulfide bonds, and then you chemically treat it, and you allow those disulfide bonds to then reform, but while the hair is curly, and so then you can get a perm, right? All right, so that's keratin. Oh, silk. Silk is another really cool, really cool fibrous protein, right? There are 3 subunits inside of silk, and one of them is all beta sheets. And these beta sheets have a really high percent of alanine and glycine residues. And the alanine and glycine, if you remember, are really small side chains. And because they're really small side chains and all of the beta sheets are packed that way, the beta sheets end up being packed super close together. And so this ends up being silk being the strongest polymer that's known. It's stronger than steel. And the really actually cool thing about silk is that not only is it strong, but it can also bend. So it can make these shapes that are required for spinning a web, but it won't stretch. It will bend and not stretch, and it is still super strong. That is very unique and very cool. All right, collagen. Collagen is found in soft tissue that holds bones together, holds muscle to bone, right? So it's a major component of tendons, of cartilage, and actually the cornea of your eye. And so it has some really long helical subunits, right? And those helical subunits are actually held together by hydroxylated, Hydroxy, hydroxylated, if I could spell, lysine residues, right? And it's important to note that these helical subunits are not alpha helices. What you should notice by looking at these is that they're stretched out and they're left-handed helices. And these are actually stabilized by interstrand hydrophobic interactions and not by hydrogen bonds. And that actually gives the collagen a lot of strength. The next kind of multi-subunit protein is a group of globular proteins. So globular proteins tend to be spherical, and they have repetitive multi-subunit inside of them, right? Excuse me. So this protein here is bacteriophage, and it's a DNA packaging motor, and it has 12 subunits. So this is like one subunit, which is equal to this one subunit, right? And so you can organize all of these subunits into basically a circle. They fit one inside the other. And so what this actually does in the bacteriophage is it assembles and then it guides newly replicated viral DNA to be placed inside of the viral capsid before the cell can lyse. And so what you can see is that any of the subunits, if I can zoom in, has 2 protein-protein interaction surfaces. So here's one, right? And here's one. Because it's going to end up being a circle, because it's going to be globular, you have to have adjacent subunits to basically interlock with one another. And so this is what the interaction surfaces allow, is that interlocking. All right, let's see. Another example of a globular protein are the immunoglobulins. Immunoglobulins, AKA antibodies. Most people know them as antibodies, right? Antibodies are an example of globular proteins. So they contain 2 copies of each related protein subunits. So you have basically a heavy chain, and a light chain. And then you have this basically times 2. And so the way that we write that is H2L2. So it's a heterotetramer. So heterotetramer, right? Hetero because these 2 are different. You have a heavy and a light. And then tetramer because you take both of these times 2, that gives you 4 subunits. So that's a heterotetramer. And so when you look at these subunits are stabilized by disulfide bonds. So you have a disulfide bond, a disulfide bond, a disulfide bond, disulfide bond, right? And let me erase that because then you can't really see it. But the job of antibodies, right, is to recognize non-self antigens. So things like viruses, bacteria, anything like that, anything that is not you, an antibody is supposed to recognize. And so you got to think that in order to be an antibody, we want to have the same shape. So we need to have some parts that are consistent from between all antibodies, and then you need to have some parts that are specific to certain antigens, so you can recognize different viruses and different bacteria. And so our variable domains are here. They're the kind of like, I don't know, jagged parts of it. And so these are the parts where you'll actually have an antibody, and I'm sorry, where you have an antigen come in and bind. So this is the antigen binding site. And when we were trying to study these, what you can do is you can actually treat them with a protease. And so not a strong protease that'll chew the whole thing up, but like a very mild protease. And so what happens when you cleave it with a protease is you end up with 2 fragments. You end up with what's called an antigen binding fragment. So here is the antigen binding fragment, or the FAB fragment, the Fab fragment. And so this contains the variable region on one end. And then you actually have this region, and this is the crystallization fragment, or the FC fragment. And this was very, very easy to crystallize. It's a very repetitive pattern, very easy to crystallize. And so that's how they kind of named them and kind of figured out their function. And that is the end, I do believe, yes, of section 2. All right, let's see if I can stop my share.